Hello, I am Ditya Shalda, currently studying BMLT in AIIMS Kalyani. So today we will be discussing about blood vials and anticoagulants. Okay. So first is what types of blood vials is there. We have blood culture bottle, red vial. You will get lavender vial, that is EDTA vial, grey vial, that is sodium chloride vial, citrate vial, and there is a special type of gel vial that is used to separate the serum and the packed cell. So this is the type of blood vials: blood culture bottle, red vial, gel vial, citrate vial, EDTA vial, heparin vial, sodium chloride vial. How you will identify that vials? So there are specific colors of every vials. Red vial, as the name suggests, it is red. Heparin vial is of green color. Citrate vial, blood culture bottle has different types of colors. Here yellow color is for pediatric patient and this orange color is for anaerobic, and this EDTA vial is of lavender color, sodium chloride vial is of grey color, gel vial is of yellow color. So next is anticoagulants. There is different types of anticoagulants used in our general laboratories. What is anticoagulant? Anticoagulant is a chemical that prevent the clotting. Okay, so first we will start from EDTA. EDTA means ethylene diamine tetraacetic acid. You have to know this full form of EDTA. We will now come to the mechanism of action. Mechanism of action, how it works. EDTA chelates the calcium ion. Now calcium ion is a uh, important ion that helps in the coagulation pathway, that helps in the clotting. Calcium itself is a clotting factor. So EDTA, when bind with the calcium and chelates it, calcium is not there for the clotting. So as the free ion is not there, clotting process is get stopped. This is how EDTA works. It chelates the calcium ion. What are the uses of EDTA vial? Most of the cases you will get that EDTA vials are used in the all hematological tests like CBC, every test, okay, hematological tests. Now HbA1c test. HbA1c test we do in the biochemistry. In parasitology also there is EDTA vial. There is, we have to maintain a specific amount of anticoagulant with the particular blood. Otherwise it can hamper the test results. Okay. So for EDTA it should be 1.5 to 2.5 mg per ml. Next come to the sodium fluoride. Sodium fluoride means that is grey vial. Along with sodium fluoride, that grey vial contains potassium oxalate. What is the mechanism of action of this using of this sodium fluoride? So sodium fluoride blocks the glycolysis. Okay, so glycolysis is a process where the glucose is broken down and it converted into the pyruvic acid. As the grey vial is specifically used for through every type of testing the sugar levels. This is the use of sodium chloride. If you can see, for RBS, random blood sugar, for FPS, fasting blood sugar, for PPBS, postprandial blood sugar. So this is very important to prevent that glycolysis process. Sodium fluoride does what? It inhibits the glycolysis, and that grey vial along with sodium fluoride it contains the potassium oxalate. That potassium oxalate blocks the calcium ion. In chloride vial, potassium oxalate is also present. So sodium fluoride is the anticoagulant used in biochemical tests like every type of sugar measurement, random blood sugar, fasting blood sugar, postprandial blood sugar. You have to know the mechanism of action. Okay, so let's come to the sodium citrate. Sodium citrate vial means what kind of vial? That sky blue type of vial. So here blood is to anticoagulant is equal to 9 is to 1. What is the mechanism of action? That works, almost every anticoagulant works in the same mechanism, that blocks the calcium ion. What are the use of sodium citrate vial? So sodium citrate vial is mainly used for PT and APTT test. Next come to the oxalates. So there are potassium oxalate and ammonium oxalate. Potassium oxalate, that is used in the gray vial along with the sodium chloride, that also bind with the calcium ion and blocks that coagulation pathway. Now if we come to the heparin. So heparin vial means what? That green colored vial. They are the anticoagulant used, heparin. What are the use of heparin? So heparin is normally used in osmotic fragility test. What is the mechanism of action? Now the heparin's mechanism of action differs from the others. Heparin binds with the antithrombin 3 and it prevents the blood clotting, enhancing the ability of inhibiting coagulation factors. And heparin also inhibit that 10A activation. In the pathway, factor 10 is activated factor 10. So heparin also blocks that. Now if we come to that red vial, it contains no anticoagulants. It just have little amount of clot activators. What are they use? Most of the biochemical tests is done with that red vial. Now come to the gel vial. So gel vial, that yellow colored vial. So gel vial contains a gel at the bottom of the tube. When we centrifuge it, that gels help in the properly separating that serum from that packed cell. What is written here? Contain a barrier gel located at the bottom of the tube. Yes. During centrifugation, this gel moves up to the interface between the serum and clot, forming a stable barrier and separating the serum from the cells. Blood culture bottle. So blood culture bottle is used in the microbiology. It contains several growth medium. It has growth medium that helps in the growth of that bacteria, so that we can detect from that blood culture. Here the most commonly anticoagulant used is SPS. Nothing more from that. So contain a growth medium which encourages microorganisms to multiply. That anticoagulants prevent from the clotting, and that anticoagulant should not be interfere with the growth of the organism. So those are about the anticoagulants. So we read about the vials, we read about the anticoagulants used in that vial. Now when you have collected blood, now you have to enter that blood into that vials. So you have to maintain a proper order of draw. This is the order of draw. Order of draw means which vial comes after which vial where you put that blood. So when you are collecting blood, that blood should be first put in that culture bottle. Okay. Then sodium citrate, then red vial, then gel vial, then heparin, then EDTA, and at last sodium chloride vial. This is very important. This you have to follow every time. That is a picture of that. First you have to collect in the blood culture bottle, then comes your sodium citrate, then comes serum. Serum means that red vial or the gel vial. Then comes heparin, then comes EDTA, then that gray vial. So this was the brief explanation about the vials and anticoagulants used in the lab.